All right, let's see if we don't run out of batteries today. Okay, guys, let's get started this morning. So, sorry last night, slides didn't come out until about 9 o'clock. That means I didn't finish your lecture until about 9 o'clock last night, too. I will send them out as soon as I finish lectures. Typically, it will be before 7 o'clock the day beforehand. Won't always be before 7 o'clock the day beforehand. Apologies if you guys didn't download or print them fast enough. I hope that everyone got my email for clickers for how to register at webclicker.org. How many of you got that email, first of all? Good. If you did not get that email, come see me, because that means you're not on my class list and I have to figure out how to get you on my class list. 2, how many of you guys were able to register successfully for WebClicker? That's pretty good. Okay, great. Great. If you weren't able to do that either, come see me afterwards and we'll talk about that as well too. 4th edition books, still at the bookstore, still on order at the bookstore. Okay. So last time in class, we talked about meiosis and mitosis, and we ended on this slide here that's really comparing these two processes. So last time we went through each one of these individual stages for mitosis, and each one of these different stages for meiosis. And there are some critical differences between these two processes. First of all, with how they end. Both are starting off with a parent cell that's 2N at the time, but mitosis ends up basically duplicating that parent cell into 2 cells that are 2N, whereas mitosis has 2 divisions rather than 1 division, and you're ending up with 4 different daughter cells, each that are haploid rather than diploid in this case. The other critical things to note for mitosis versus meiosis, in my opinion, is the way that chromosomes line up at metaphase versus metaphase 1. So remember, in mitosis, all the chromosomes are lining up and they're not necessarily pairing with their homologous chromosome. But in metaphase 1, they are pairing with that homologous chromosome. So mom's chromosome and dad's chromosome, with those 2 sister chromatids each, are pairing at that metaphase plate right there. And that creates one of the important things that meiosis does, which is crossing over and recombination. This basically segregates different variants along a chromosome. It is incredibly important for sexual reproduction, adaptation, and any kind of evolution. So let's go through that process a little bit more. I showed this slide before. This was maybe the second slide before we ended right there. And what this was basically trying to show you is that there are both single and double crossover events in that metaphase 1, where it's not only those chromatids that are lined up right next to each other, but in reality, instead of these cartoon diagrams, they're kind of all interlaced all over each other. And they can actually cross over between multiple different sister chromatids there. And that creates this amazing amount of kind of variation in those gametes, where you can see that variants along these different parts of the chromosome may be from mom and maybe from dad. And by crossing these together with a male and a female, we can produce a lot of different variation in a few number of generations. So, how many crossing over events actually happen in a single generation? I'm not sure if I told you this number or not, but it's somewhere between 50 and 100. And it differs depending on if you're in, this is in humans. It differs whether you're a male or you're a female and where this is actually taking place at. So other factors that may come into play, because we're talking about humans here, you have 23 chromosomes and we have a certain size genome. But if you can think about other organisms or other species, this may differ dramatically because you have different numbers or different lengths of chromosomes. Okay. So let's talk about how these individual genetic variants on a chromosome actually segregate out. This is just straight out of your book here, but it's showing something that's important, and I want to make sure that you get the main message here, that 2 different genes on the same chromosome can segregate from mom and dad because of these crossing over events here. So we have those original chromatids. They duplicate during the S phase. They become sister chromatids linked at that centromere right there. And on those different chromosomes, during prophase 1 in meiosis, they can have those crossing over events that may swap out different parts of those chromosomes right there. This is a single crossing over event right on those, just like they normally picture it in cartoons, right on the 2 middle sister chromatids. And then showing exactly as before, we're segregating out those 2 different genes
with different alleles, and now we've mixed up the alleles for mom and dad. So if this red is mom, we have both of the original alleles that mom possessed in this gamete, but we have one of mom and one of dad's alleles, and the second gamete, same thing in the third gamete, and all of dad's gametes, or all of dad's alleles in the fourth gamete. So not only do we segregate out different alleles or different genes on one chromosome, but meiosis is also important because we can segregate chromosomes as well too. And this one might seem a little bit more obvious even. If you have 3 different chromosomes here, all that are pairing up this way, the way those things align at metaphase 1, they may align in this orientation, where all of moms are on one side and all of dads are on the other. Or they could have any of these other different orientations. This is just with simply 3 chromosomes here, where you get pairs of mom and one of dad, pairs of dad and one of mom, or even this kind of opposite, where you get different chromosomes on those sides. But we come up with all of these different combinations to segregate these different chromosomes too. And this is at random as well. There is no outside bias that we know about in segregation of chromosomes. Okay, so you can imagine how you might create a formula for any number of chromosomes that you could actually have here. And that equation would be 2 to the n power. So say you have 2 different chromosomes, 2 to the 2nd power, you could have 4 different combinations that you could segregate out as gametes. If you have 3, we obviously 2 to the 3rd power is 8, 2 to the 4th power would be 16. You can imagine that you get these combinations, and as soon as you have 23 chromosomes, you have a lot of different combinations that you could actually get in any one of those different gametes. Okay. So, quick comparison between mitosis and meiosis. Again, this is straight from your book. Both involve cell division. Only meiosis involves those chromosome reductions. You go from 2n to 1n in the end. You don't produce genetic variation in mitosis, although this is kind of a little bit of a lie. If you look more into depth, introductory textbooks won't tell you, but there is some crossing over events that happen in mitosis as well too. But that's not important for this class. I won't ask you a question on it. There's crossing over in meiosis, but not mitosis. As I just talked about, there's that random distribution of maternal and parental chromosomes that depends on the number of chromosomes you have for the amount of combinations that you can actually make. And as I said, the 2 biggest differences are in metaphase and in anaphase, with how the chromosomes line up in metaphase versus metaphase 1, or how it's either chromatid separating or homologous chromosomes separating in anaphase. So these are kind of the critical details that I kind of expect you to know to differentiate these 2 processes. Okay. So in these germline cells, in the gonads or the ovaries for humans, is it just meiosis that takes place? No. There's mitosis in those germline cells too. There isn't meiosis in somatic cells, but there is mitosis in germline cells. All right? Not a human biologist, so my names might be a little tricky here. And I don't think the terminology is actually that important. For somatic gonad, I'm going to let you read that up there. Try this one more time. The flowering ones I'll have down, I promise. Spermatogonium. Spermatogonium. Spermatogenesis, but spermatogonium are the diploid cells that we start off with in the gonads that actually go through meiosis to produce those haploid cells originally. But the critical part after that is that there's actually some mitosis after that in not necessarily the sperm cells, but in the ovaries here. Am I correct on that? Okay. I'm going to advise you guys to read the book on the human side of this part of this process. But mitosis actually occurs in, as well as meiosis, in the ovaries and in the gonads. Okay, so the plant side, we got this one a little bit better down over here. So remember your plant life history over here. The flower is actually a diploid tissue right here. In that flower, which can be a hermaphrodite for many species, we're going to have meiosis in the anthers to produce pollen cells. These are called microsporangium. And then we have also meiosis happening in in the ovaries and plant tissue as well, in the style. And those are called megalosporangium. As those two things come together after meiosis, they can fertilize the same plant. In some plant species and other plant species, there's self-incompatibility mechanisms that don't allow self-fertilization. They produce that single zygote, which is a seed, and that results in mitosis, going back through the life cycle again. The important part in plants is, again, that in both the anthers and in the ovaries, there is meiosis followed by mitosis. And that mitosis is incredibly important for plant cells because actually in a single pollen grain, you have two different nuclei that are going down the pollen tube to get to the ovary. One of them is controlling the direction of that pollen grain, and the other one is the one that actually fertilizes the egg cell within the egg within that ovary. And the same thing happens in the female tissue there as well. They actually go through 4 different meiotic events following meiosis. So you have a lot of these that actually kind of crap out along the way. So you only keep one of the products of the 4 from that original meiosis, and it duplicates multiple times into different functional tissues. Okay. That's meiosis and mitosis. Today we're moving on to Mendel's principles of inheritance. Today, I'm guessing, is going to be mostly review for a lot of you guys for a lot of these different terms. I'm guessing you've had this again in high school and somewhere in one of your first 2 classes that are here. But I think it's really important that I give you some necessary terminology because I'm going to be throwing those words out for the next 3 months, and it's going to be fast at different times too. So we got to get these in your head. We'll go through a monohybrid cross. We'll go through how we proved Mendel's hypotheses about that monohybrid cross and the different factors that were involved in producing the different phenotypes in the following F1 and F2 generations, how he showed that that was actually correct. And then we're going to do some probability and statistics in here. And then we'll do a quiz for the last 15 minutes, basically on history of genetics, a couple things, and basically meiosis, mitosis. So, if you guys notice that it is 9:35 and I'm still talking, somebody please raise their hand and stop me from talking. Okay. Modern terminology. So Mendel called genes actually factors, but in this class we're going to say a gene is an inherited factor that helps determine a characteristic. It's a region of DNA. These characteristics don't necessarily need to be traits that are manifest physically. They can also be the creation of proteins or enzymes that are critical for function. Those genes, each with mom's copy and dad's copy, if they differ in at least one base pair, they're referred to as different alleles in a sense. So 1 or 2 more alternative forms of a gene. And remember, alleles in this case have to have some genetic difference between them. The locus is the location on the chromosome that's occupied by that gene and those 2 alleles. So the locus is basically the address on the particular chromosome for that given gene. So you will hear things later on in class. I will say one of the things I work on is this phenotype called cyanogenesis, the ability to produce hydrogen cyanide. It's been known since the 1920s, and since the 1940s it was referred to as the AC gene. Great, we referred to that all the way until 2007 or 2008, when somebody actually discovered that this AC gene, this Mendelian factor, had a molecular genetic basis that was a particular gene that was already named. It was called CYP79D50. Great name, yeah. AC was a lot easier. And turns out that gene was a complete gene deletion as well. We'll have a complete conversation on this, but you can see that there's multiple levels of terminology that we have here. When we talk about the Mendelian genes, often we talk about simple things. AC, R for wrinkled versus smooth peas. But remember, those have a molecular genetic basis too, as does an allele. Okay. Genotype is a set of alleles possessed by an individual organism. So this particular pea plant right here has a genotype of big R, little r. Heterozygotes, an individual organism that possesses 2 different alleles at a single locus. Again, these 2 sister chromatids right here are heterozygote. If you had just 2 big R alleles, you would be a homozygote. And for our practical purposes, when I say phenotype, trait, characteristic, or character, I'm not going to, most of the time in modern terminology, people don't differentiate between these different traits here. And I won't be quizzing you on it or testing you on it. I could say something's a character, I could say something's a trait, I could say something's a phenotype. But most people, when they use those, they're interchangeable with one another. So, that first lecture, I gave you one slide on Mendel, which is not doing it fair justice, and we will now give it probably a lecture and a half of what on Mendel. And all I told you before, when we were talking about Mendel, was we looked at this green versus yellow pea color, which is what we're going to call the monohybrid cross today. Mendel's major conclusions, Mendel, again, remember, was this Austrian friar that now is, that was at a monastery, and actually was a very well-educated friar at that monastery, too. He did go to a semblance of college as well, and he had a really good concept of experimental design in the scientific process, which we'll talk about in a couple of minutes here. But his two major conclusions that I told you about before out of this monohybrid cross was this principle of segregation. We'll talk about beginning here. Each diploid individual possesses two alleles for a given locus that segregate when gametes are formed. This is exactly what we were talking about in mitosis and meiosis earlier, except Mendel didn't know anything about chromosomes at the time. And then we have the concept of dominance. When two different alleles are present in a locus, organisms will exhibit the phenotype of the dominant locus over the recessive locus. Okay. So, what traits was Mendel actually using? Mendel was using Mendelian traits. These traits that are discrete traits that have been renamed, and people often use the term Mendelian to describe them. So there are things that are very discrete in terms of endosperm color, whether it's yellow or green, seed shape, round or wrinkled, seed coat color, pod shape, pod color. You notice each one of these things have a very discrete phenotype that you can observe in a cartoon very easily. In practice, these things aren't quite falling into those discrete categories. As we all know, what's yellow as compared to what's green can differ between individuals as well too. So there is some level of quantitative variation within each one of these qualitative characters that they have. But Mendel is ignoring that quantitative variation when we talk about this here. Now, other things, also discrete characters, flower position, like at either the apical meristem or at a lateral branch over here. Other things we often consider more quantitative traits, like the stem length right here. Although Mendel was using two different varieties, one that had an extremely long and one that had an extremely short, again, there's clearly quantitative variation within those different stem lengths. And one thing that's important to remember about phenotypes is we put this in perspective of the different categories of phenotypes, where we have a continuum from these discrete phenotypes to these quantitative phenotypes, like human height, those famous pictures you always see with people lining up in normal distributions based on their size that they actually are. Remember, to get a particular phenotype, we have contributions both from genetics and from the particular environment that you're in. In terms of these Mendelian discrete phenotypes, what Mendel is talking about, these are completely genetically controlled in terms of the qualitative variation he's talking about. But there is also clearly environmental phenotypes too. If I take one of Mendel's, I take plants that are both yellow and green in pea color, and I stick them in a temperature that's 4 degrees Celsius, something that's very cold for them over a long period of time. I'm going to make both of these turn red, and that'll completely change the phenotype here. And that's based on environment that you have. Anything that changes via environment is a plastic phenotype that you have for this variation. And of course, there are things where you can get the interaction between these two phenotypes too, gene by environment interactions. You may have one particular gene that allows you to perceive a cue of some sort and create a neural phenotype. That's a bit too detailed for today's lecture for going into G by E interactions. We will go into it a little bit later as well. Okay, so what was Mendel's experimental design here? Important to know out of this is Mendel that pea plants are hermaphrodites who don't have any self-incompatibility barriers here. They can reproduce with themselves very easily. And in fact, we do this all the time with different plants. This is one of the big advantages of having plants, because you can't cross humans. So simply, you can take an anther that has a bunch of pollen grains in it, smash it up, and use a paintbrush or forceps to put it on those styles. That will result in a cross, and you actually get offspring out of this. So when we're talking about this experiment, if we have a simple phenotype, you have yellow versus green seeds. Remember when Mendel was actually creating these hypotheses back in, let's say, 1866 was when his paper was actually published. One of the major things that was thought at that point was this phenomenon of blending inheritance. That if you take mom's phenotype and you take dad's phenotype, what you should get is something in between of that. And that works fairly well for some of those quantitative phenotypes that we were talking about. But this is how we show that that's not actually the way that things work at a genetic level. So the hypothesis for this blending inheritance with Mendel's experiments would be that we got something that was in between these two seed colors when we crossed a parental plant that was yellow and green. Okay. This is what Mendel actually observed when he looked at all of those different phenotypes within this crossing scheme that he had. So, important to note before this as well, he bred these two parents for multiple generations beforehand to make sure they were homozygous. If you just grab two pea plants out in nature and brought them into the lab to do these crossing experiments, there's a fairly good chance that you're going to be bringing in heterozygotes into the lab right there, and you wouldn't have this monohybrid cross design coming out of that. So this is that's an important consideration to think about here. When he crossed those two, the wrinkled and the round seeds together, he got an F1 generation that was all round seeds. Then he self-fertilized that F1 generation, because they're hermaphrodites again, and what he got out of it was 3/4 round, 1/4 wrinkled. In fact, he did this experiment many, many times, and clearly he got something that was fairly similar in proportion to this. And we'll talk about how we can actually statistically determine if these frequencies match these frequencies at the end of this particular lecture. Now, from this, he hypothesized these genetic factors. Oh, sorry, animations are going on. Okay. He hypothesized that this is what was occurring here. That he had these two genetic factors, one was a dominant factor and one was a recessive factor. He designated them by using capitalized and small letters over here. So he thought that if we had two copies of each one of these genetic factors, you bring them together, they segregate independently. So there's an equal chance that this R goes with that R, this R goes with that R, this one with that one, this one with that one. You can do it like a little foil diagram out here. If you cross those two things together, we had to be getting at least one big R from dad and one little r from mom. So all those F1 seeds were going to be heterozygous plants, one big R allele, one little r allele. However, if you cross those heterozygotes together, there's a chance that you get big R, because your mom and your dad both could inherit that dominant allele, and that will happen 25% of the time. There's a chance that your dad could have that dominant allele and your mom could have the recessive allele, or vice versa, where your mom has the dominant allele and your dad has the recessive allele. Or you could inherit both the little r's from mom or from dad. And all four of those possibilities occur at an equal frequency because of the independent segregation of chromosomes that we have. So this was his hypothesized frequencies down here, pretty close to what he actually found. When you look at the phenotypes of those, obviously all three of these, because this is the dominant factor here, would be round seeds, and the fourth one over there would be wrinkled seeds. Now, okay, what are the actual results that we have from that? This is the longer definition that the book gives you for the principle of segregation. Basically, diploid organisms possess two alleles. They segregate when the gametes are formed, and one allele goes into each gamete. And that concept of dominance, that there is some sort of dominance, that one allele is seemingly dominant over another. Two alleles are present, only the trait encoded by one of them, the dominant one, is observed in the phenotype. This dominance in his traits was complete dominance. And we will talk about incomplete dominance tomorrow. So, this is all well and good. All of that monohybrid cross fits his hypothesis for what he thinks was happening with these two different genetic factors, with those two different alleles and those independent segregation. But how can we prove that this is actually happening? Or how can we actually test this hypothesis that we have these genetic factors occurring. And that's how you come up with this test cross, where we cross a heterozygote, say from those F1 individuals, with the recessive parents. And we can predict what frequencies we should get out of that cross and see if we actually get something that matches our expected ratio here. So again, this is the tall versus the short plant rather than the wrinkled versus round seeds. We can all do a Punnett square over here, where you put dad's gametes on the y-axis, mom's gametes on the x-axis. Because they're independently segregating, each one of these boxes should have equal frequencies there. And we simply can put these two together here. So you get big T from dad, little t from mom. And what you should get out of this is that half the plants have that dominant phenotype, and half the plants have that recessive phenotype. So this is coming out as something different from that 3-to-1 ratio we observed in the F2 generation, and can confirm what Mendel was talking about when he was looking at these genetic factors. Same thing happens with the round versus wrinkled peas. I'm not going to go through this exact thing again, but so let's talk a little bit about probability and how can we actually test if those observed ratios that Mendel got actually correspond to the expected ratios that he would get from his model of independent segregation. So probability is a measure of likelihood that an event will occur, how often a pattern exists via random chance. So we can calculate probability typically by the number of events at times the event is actually observed divided by the total number of events that it occurs, if we're doing this in a very empirical concept. If you flip a coin 100 times, you might not get 50-50 out of it. If you flip it infinite times, you should get, with a fair coin, 50-50 heads and 50-50, or 50% heads and 50% tails. There are two different types of probabilities that we can create. We can create these empirical probabilities that are determined by observing a large number of trials. If I was flipping a coin 100 times and trying to figure out if it was a fair coin from that, that would be an empirical probability, because I'm observing a large number of trials and looking at the results from that to create an expected ratio. So, for instance, possibly a better example is the probability of tomorrow being rainy. So if we observed year after year after year on January 26, we could infer some frequency of temperature, some frequency of precipitation. We could add to that probability estimate by looking at the days beforehand too, and we can create different expected probabilities for the probability of tomorrow being rainy. In fact, this really what meteorology actually is looking at the past to infer the future, and looking at climatic events around the country to actually infer weather. But we also have a separate kind of probability, and that's these a priori probabilities that are determined by the nature of the event together. So if you have a fair dice, every one of those 6 sides of that fair dice should have an equal probability of 1/6 of coming up when you roll it. Drawing any kind of card from a deck of cards, you have 52 cards there. The probability of drawing one card from that is 1 out of 52. Or, more importantly in this context, thinking about this like genetics, the chance that an offspring of self F1 heterozygote will be either 25% recessive, or will be 50% heterozygous, or 25% with dominant homozygous in the progeny generation. I'm going to go through 2 rules of probability here, and we'll go through one more rule of probability when we talk about dihybrid crosses. So the first rule is this product rule. It's the probability that independent events that occur in concert is the product of their separate probabilities. So if we're thinking about this in terms of that monohybrid cross that we just had there, that are heterozygous for P shape. They have one big R allele and one little R allele. What is the probability that offspring will be homozygous recessive? So the probability that you get an egg from that F1 that have a recessive allele, there are only these 2 chromosomes within there. So if you're going to, probability of pulling one or the other is 1/2. The probability that you get a sperm from the F1, again, you only have those 2 alleles on 2 chromosomes. They segregate independently in meiosis. The probability of that is 1/2. Therefore, the probability that both of these two events occur together is going to be 1/2 times 1/2 equals 1/4, or 25%. That's your product rule right here. If you multiply two different probabilities together, that's the chance of both events happening together in concert. Okay. The additive rule, the probability that one of several mutually exclusive events will occur is the sum of the probabilities of these individual events. So, in a monohybrid cross, again, heterozygous for plant height this time, big D and little d. There are two ways that a heterozygote can actually be produced. The dominant allele and the recessive allele. Or you get the dominant allele in the egg and the recessive allele in the sperm, or vice versa. The probability that the offspring will be heterozygous then is the sum of both those probabilities. So we just talked about right now the probability that you get a big D allele in the egg with a little d allele in the sperm would be 1/2 times 1/2. You have two chromosomes there, they segregate independently. So you get 1/4. Again, the probability that big D in the sperm and little d in the egg is another 1/4. But, so if we add these two probabilities together, these are the two options that could potentially happen here. So the probability of getting a heterozygote would be either this or that. Simple addition, half the time we should get a heterozygote here. How does Mendel actually know that his ratio matches a 3-to-1 ratio? How many of you guys have done a chi-square test? A couple. When did you guys do it? Did you do it in high school? Yeah? Did you do it in college? In a lab or in a different class? First year?
Different class.
Different class, okay. Okay, so how does Mendel know if he's getting a specific ratio? Does this essentially this observed ratio that he gets when he gets these 5,800 to whatever 1,800 T's actually deviate from the expected ratio? The way we're going to test this is what's called a chi-squared goodness of fit test. This is your test distribution, your test statistic right here. It corresponds to a chi-squared distribution, which we'll talk about in 1 second here. This is simply the sum of all of your different observed events minus expected events squared. So it doesn't matter if you're above or below the number of expected events that you actually have. Those two things become the same in this because of your square divided by the number of expected. Okay, so this is an experimental test that only works with count data. And what we're looking at here is something like this. If we're thinking about heads or tails, the counts of heads versus tails. The probability of heads or the probability of tails you get is 50%, essentially, from an expected, our kind of expected ratio. As in the context of our genetics, the count of dominant versus recessive, we expect that we're going to have that dominant phenotype 3/4 of the time, and the recessive phenotype 1/4 of the time coming out of that monohybrid cross, just to create expectations before we look at the observed value. So that chi-squared is a statistic. It's not actually the probability here. We can match it to a particular distribution and understand what the chance of getting that is compared to if we just took random data by chance with that particular expected ratio that you have. So another important topic in this that you're going to need to understand the chi-square distribution is the amount of degrees of freedom you have. So for instance, if we flip a coin right now, you have the probability of getting heads or tails. If you get heads, you cannot get, or if you don't get heads, you can't get anything else besides tails. Thus, that event only has a single degree of freedom versus multiple degrees of freedom. And we can use this degree of freedom and the value of chi-squared to determine this experimental results, and if it's outside a range that we would expect from chance with that particular number of samples that we actually have. The results, let me go here. I lost my distribution in here. Sorry guys, okay. Let me wait a little bit.
And just to let you know, it's 9:34.
Thank you. Okay. We'll come back to chi-squareds in the next lecture, but I want to make one point before I hand out this quiz right now. I'll come back to this diagram. How many people cannot see this diagram at all? Okay, we'll come back to this in the next lecture too. We have a mean around this particular distribution. So, if we have a particular expectation for this given phenotype, say it's the average right here, and we have a certain number of events that we could do by chance. Say we flip a coin 20 times, 30 times, whatever else. There's a chance that we're going to get 9 heads, 8 tails, or I'm sorry, 9 heads, 1 tail in here. There's a chance we're going to get 5 and 5. Your average would be centered around getting 5 and 5 for that 10. But if you do this a bunch of different times, we're going to get all sorts of different things, ranging from 1 to 10 times that you get heads in here. And we can create an expected distribution. So if you fall in the outside of this particular phenotypic distribution in the bottom 5%, we'd say that would deviate from chance. So essentially what we're looking at for statistical significance is this alpha value, where we're looking at something that's not a false positive, essentially, here for a signal. We're going to have to talk about this a lot better when I'm not supposed to be giving you a quiz. We'll come back to this next lecture. We didn't quite make it to the quicker question. Sorry, guys. Okay, instructions for this quiz. When you finish it, let's stick it on that table right by that door out there before you head out that door, because there's a lot of people coming in this way. One pass back.